There's been a revolution in cancer research. I will argue that we can only understand this cancer revolution by thinking broadly about the origin of novelty in biology. And working in the other direction, I will also argue that cancer research teaches us a lot about evolutionary novelty. So to analyze this synergism between cancer and novelty in biology, let's turn first to evolutionary aspects of novel traits. Darwin's great book, The Origin of Species, could have been called The Origin of Novel Traits, because Darwin was interested in one of the most profound puzzles in all of life, which is, where did all of that diversity that we see in life, where did all of that diversity come from? Which is just another way of asking, how do novel traits arise in biology? For example, humans stand upright and walk on two legs. Our closest ancestors, chimpanzees, gorillas, and orangutans, they typically walk on all four limbs. So how is it that humans came to become permanently upright and walking on two legs? Now, the usual way of describing the evolution of such a complex trait, which requires changes in bone, in muscles, and in behavior, and all of those changes happening in a coordinated way, we usually think of such complicated traits as arising by a sequence of genetic changes. For example, there might be a mutation or a rare pre-existing genetic variant, and then as the environment changes, those genetic variants become favored and spread through the population. And we usually think that a single genetic change is not sufficient for such a complicated new trait to arise. And so we imagine a sequence of mutations and genetic changes originating and spreading through the population. And eventually all of those genetic changes together coordinate to make this complex new trait. And this is the classic view in evolutionary theory. It's widely accepted. And we can call it the genes first view, the idea that first genetic variants arise, and those genetic variants spread through the population, causing phenotypic change in the trait. Now, there's an alternative view that's been developed recently called the phenotypes first view. And this has been developed most extensively by Mary Jane West Eberhard over the last 20 years. And I can introduce this with a story. In the 1940s, there was a goat that was born, and its front legs were malformed. And so this goat, from very early in life, learned to walk on its two hind legs in an upright manner, quite similar to the way in which we walk. And as that goat developed, the forces acting on its bones and muscles caused changes in its bones and muscles so that it was quite different from a typical four-legged goat in the way it was put together. And when that goat died, a Dutch anatomist dissected out the goat and looked at its bones and its muscles and so on. And that person did indeed see that the bones and muscles had changed a lot in this particular goat. And although they were not identical to the way in which our bones and muscles look, they were quite a bit like ours when compared to a typical goat. And so we can see that the forces that were acting on these bones and muscles during development caused the goat to respond in a phenotypically flexible way to those novel forces, changing the way the individual was put together. And that's not completely surprising. We know that when we change the forces acting on our muscles and our bones, that our muscles and bones remodel themselves in response to those different forces. So there's intrinsically a lot of plasticity and flexibility in the way in which individuals develop. And Mary Jane West Eberhard in particular has emphasized with regard to this goat and with regard to novel traits that the way in which they might first arise is in this sort of phenotypic flexibility. So in this particular case, we have bones, muscles, and behavior all changing together in a coordinated way. And this makes sense because development is flexible and responds to changes in the individual and in the environment. And in this particular case, there was parts of the normal developmental program for bones was changed, parts of the normal developmental program for muscles was changed, and parts of the normal way in which behavior develops in an individual was changed. And so West Eberhard calls that developmental recombination. You combine bits of different developmental programs in novel ways, and you can in one step get a coordinated complex trait arising. And we know this can happen. So remember this concept of developmental recombination, because we're going to revisit it later when we talk about cancer. Now there's one problem with this argument with regard to the origin of novel traits, which is that there were no genetic changes, and therefore the trait is not heritable. It doesn't get inherited and passed on to future generations, and a trait that's not heritable has no evolutionary consequence. So how can this be important in evolution? And the idea is that first these phenotypic changes arise, phenotypes first, and then if there's a novel environmental challenge in which this phenotypic variant is favored, then that phenotypic variant or trait will arise in each generation as individuals respond to the novel environment, and that over time there can be genetic changes that stabilize and modulate that novel phenotypic expression. And once those genes arise that stabilize the phenotypic expression in this new way, then the trait becomes heritable. So it's phenotypes first and genes follow, as West Eberhard has emphasized. So that's the first part of the talk, and where I've talked about the distinction between novel traits arising by genes first and phenotypes following. This is the classic view in evolutionary biology and in cancer. And then there's this alternative view, which is that phenotypic responsiveness produces new traits first, and the genetic changes happen second. And so the question is, how does it really happen? And the answer is we don't know. I'm sure that both processes happen occasionally in nature, but what we don't know is how important, the relative importance of those two different approaches. And this is where cancer comes in with regard to this general question, because cancer actually provides a lot of insight into this genes first versus phenotypes first problem or contrast. And so we'll talk now about cancer. And I said at the very beginning of the talk that there's been a revolution in cancer research. And this revolution in cancer in the last few years, in cancer studies in the last few years, has arisen because of novel techniques that allow us to measure things that we could not see before. Previously, if you wanted to measure the genetics of some cells in an individual or in a tumor, you had to take a lot of cells. Millions or even billions of cells had to be put together and mixed up, and then you would get enough DNA so you could measure the genetics. But in that sense, you're measuring the genetics of the aggregate of cells, and you cannot measure the variation from cell to cell. And in the same way, if you want to measure what kinds of proteins are in cells, then you had to put together a lot of cells and then measure the aggregate protein. You could not see the differences in the protein expression in the different cells. But in the last few years, it's now possible to measure at the single cell level the genetics and the protein content of individual cells. And so now we can actually see the variation between cells. And it's that variation between cells is actually the scale at which changes happen during cancer. So for the first time, we have the resolution to see what really changes as cancer develops. And this has been revolutionary, I would say. And since it's just starting, we'll continue on for the next few years. And to give you some idea, let's first talk about one important problem, which is how do tumors evolve resistance to chemotherapy? So a person, when they get diagnosed with a tumor, with a cancer, and they go to the clinic, then they often get treated with chemotherapy, with drugs. And typically the drugs work at first. They knock the cancer back and cause a remission. But over time, the tumors almost always evolve to be resistant to the drugs, and then the tumor begins to spread again. So the real challenge clinically is the evolution of resistance by the tumor. And the evolution of resistance is really the origin of novel traits to an extreme environmental challenge. From a tumor's point of view, drugs are an extreme challenge that will tend to kill the tumor, kill the cells. And so the cells must evolve to overcome that extreme challenge. And to do that, they typically either evolve novel traits or express their existing traits in a novel way. And the standard view of how they do that is that when you treat with a drug, there are pre-existing genetic mutants in a small number of cells. And so the drug kills most of the cells, but a few cells survive because they're genetically distinct. And those genes then spread because now those are the only cells that can survive in the environment of this drug treatment. So this is a genes first view, and this is almost the universal view in how resistance evolves to drug treatment, as it is the universal view within evolutionary bio in general for the origin of novel traits. But recent studies have shown that maybe it's not always that way, that maybe this phenotypes first idea actually explains the evolution of resistance in some cases. Now we know in some cases that the genes first process really does happen. There's been studies of a particular kind of breast cancer where people look very carefully at how resistance to the drug evolved that was being used to treat this breast cancer. And it was pre-existing genetic variants that were already present before the drug was used. So most of the cells died, they were susceptible. The resistant cells that were genetically distinct spread. Classic genes first view. But recent studies have shown that phenotypes first actually happens at least occasionally, if not frequently, in the evolution of drug resistance. And to understand why this is interesting from a phenotypes first point of view, let's back up a little bit and think about what this revolution in cancer research allows us to measure. In particular, what's being measured most often are the proteins that are contained within an individual cell. And this is important because the proteins define cell state, what kind of cell it is. Your skin cells make skin proteins, your bone cells make bone proteins, your brain cells make brain proteins. Different types of cells in your brain make different kinds of brain proteins. And so basically you can look at our cells as having different cell states according to their function. And those cell states are defined by the complement of proteins that are carried within the cell. And we can now measure at the single cell level or estimate the kinds of proteins in different cells in the cell state. And this is important for both cancer and also understanding how our bodies normally develop. So when you're born, you start life with a single cell. Your father's sperm and your mother's egg unite to make a zygote. And from that one cell, your body develops to produce about 50 trillion cells, which is about how many cells your body has. And those cells differentiate into many different kinds of cells, skin, bones, brain, and so on. And as that developmental process is happening, the cells are changing state. They're changing the set of proteins that are being expressed through a sequence of cell state changes. And we can now measure those cell state changes, and this is very interesting. And it's also interesting because in cancer, we can see the cell state changes. So for example, let's talk about the evolution of drug resistance in melanoma. Melanoma is an interesting kind of cancer. It develops from the skin, from pigmented cells in the skin called melanocytes. And melanoma develops a particularly aggressive kind of cancer, whereas other skin cell types that develop cancer are not so aggressive. And so one interesting question is, why is melanoma so aggressive? And the reason is that melanocytes are a particular cell state, including expressing pigments. And as melanoma develops from melanocytes, the cells tend to de-differentiate and go backwards in the developmental hierarchy of cells that made those melanocytes to a more primitive, less developed cellular type called a neural crest cell. And neural crest cells are very important in development. In early development, neural crest cells move around a lot in the embryo and in early development to help develop different tissues. They're kind of organizers of development of different tissues. And these cells are very mobile. They're very good at surviving in different kinds of environments in the body. They tend to be fairly resistant to stress and so on. And when melanocytes de-differentiate into neural crest-like cells, they then become very mobile. And this is what makes a cancer very dangerous. Tumors that can spread rapidly through the body. And so as melanoma becomes neural crest-like, it takes on this rapidly spreading tendency. And so we see when we treat, there's been studies recently that show when we treat melanoma with drugs, in a particular study what happened was that the melanocytes and the neural crest cell-like cells exist in the tumor. And there's a particular protein, a transcription factor called MITF. And MITF, the abundance of that protein within cells fluctuates randomly within cells. It's only a small number of copies of the protein in a cell, and that number tends to go up and down, more or less randomly. And when the cells express a lot of MITF, then the cells are melanocyte-like, they're differentiated. And then when they express a small amount of MITF, they become more neural crest-like in their cellular state and the kinds of proteins that they express. They become more mobile, they become more resistant to stress, they survive better under different conditions. And when we treat with a drug, what we see is that some cells are fluctuating and in the neural crest-like state, and those cells tend to survive the drug treatment, whereas the cells in the melanocyte-like state tend to die. So some of the cells survive because they just happen by chance to be in a neural crest-like state. But over time, those cells the MITF is fluctuating, and some of those cells will then express and become more melanocyte-like as the MITF goes up in some cells. Those cells become susceptible. So it's a non-heritable form of resistance because the neural crest-like state is not stabilized and heritable from cell generation to cell generation, but just is fluctuating. But over time, what we see is that some cells get into a neural crest-like state and are resistant, and then there's secondary changes in the cell that stabilize that characteristic, epigenetic changes. And once that happens, the cells become stably neural crest-like and stably resistant, and that causes resistance then to spread, and the tumor is then resistant to that drug treatment. That's a phenotypes first pattern, because what happens first is that the cell phenotypes are fluctuating and they're not heritable and there's no genetic change. And it's only secondarily that the stabilization by epigenetic or genetic changes happen that make that resistance heritable. So it's phenotypes first. Now we can talk about a second example of the evolution of drug resistance in ovarian cancer. And in this study, the investigators took ovarian cancer cell lines and put them into petri dishes initially. They also later did mouse studies, but initial study was in petri dish with tumor cell lines. And they put some drugs chemotherapy drugs into the petri dishes with these cells. If they put a high dose of the drugs into the petri dishes, the cells die. But what they did is initially they started with a low dose of the drug. And when they use a low dose of the drug, then some of the cells survive. And then they treated for a while, say a couple weeks, and then they doubled the dose. Same thing, and then doubled the dose. And they repeated this, doubling the dose many times until they eventually got to a very high dose that would have initially killed the cells. But what happened over that time was that the cells were sequentially evolving resistance to the increasing doses. And they measured the cell state changes within those cells, the cancer cells. And what they saw is that the cells went through a sequence of 5 different cellular states with different complements of proteins. And those cell state changes actually recapitulate what's a transition from a differentiated type of cell to a de-differentiated type of cell. In particular, the ovarian cells are epithelial cells. Epithelial cells are things that are on the surface of your body. Your skin's epithelium. The lining of your lungs is a surface, it's epithelial. The lining of the ducts in the ovaries is epithelial. The lining of the pancreas tissue ducts is epithelial, and so on. So many of your tissues have linings that are epithelial. And most human cancers, not all, but most human cancers are epithelial, come from epithelial tissue. And what happens is the cells de-differentiate. They de-differentiate towards what's called mesenchymal type of cell. So this is called an epithelial to mesenchymal transition, which is well known in cancer. Seems to be very typical of later stages of cancer progression. And what's happening is that the differentiated epithelial cells, which tend to be fairly stable, don't move very much, and are sort of terminally differentiated, de-differentiate back to a more primitive developmental type of cell. So mesenchymal cells are very important in development. They tend to differentiate into many different kinds of tissues, so they're phenotypically flexible. They're more resistant to stress. They can survive under a wider range of conditions than most epithelial cells can. And so what they observed was an epithelial to mesenchymal transition through a series of cell states. And that was phenotypic changes without necessarily any genetic change. And then later on, they saw the stabilization of that mesenchymal state through epigenetic changes, which made the changes heritable and allowed the cells then to become permanently resistant to the drug treatment. So once again, we see a phenotypes first pattern for the evolution of drug resistance. And now what we'll do is turn to the progression of cancer itself. How do cancers actually form? And the evolution of cancers within the body is actually the evolution of a sequence of novel traits, because cancers take on a variety of novel characteristics that allow them to spread throughout the body and survive under different conditions and become aggressive. And so we actually have then a sequence of cellular and tissue changes, which are novel traits, which allow tumors to evolve and develop to be aggressive. And so this is very much along the lines of what we're discussing. And traditionally in cancer, when we talk about the evolution of cancers, we think about classic genes that drive cancer evolution: KRAS, P53, the retinoblastoma gene. And the typical view of cancer evolution is that you get mutations in these genes first, and those mutations drive the progression of the tumor. That a KRAS mutation causes cells to divide more rapidly, and of course rapid cell division would be a characteristic of a tumor. That P53 mutations tend to knock out the normal program within your cells, which causes your cells to die when they're behaving abnormally. And this is important because as cells progress towards cancer, P53 tends to cause the cells to commit suicide, to die. It's a programmed cell death. And in order for a tumor to spread, you have to knock out that programmed cell death. And that happens through P53 knockout mutations. And so the typical view is you get a sequence of mutations, genes first, which drive the progression of the cancer. But now that we have the ability to measure at the single cell level and we can measure cell state, we're seeing a somewhat different story. And so I'll give you a couple of examples of that to illustrate how the view of cancer progression is changing really just in the last couple of years. And the first thing we'll talk about is lung cancer. And in this particular study of lung cancer, what happened was that first a KRAS mutation arises. And KRAS, as I said, is thought to be associated with an increase in cell division. And KRAS mutations early in lung cancer development often lead to small tumors developing, but they're benign. They don't actually spread. Their spread is limited. So they cause a bit of a little growth, tumor forming, but not an aggressive tumor. And then we know, and this has been known for many years, that one of the next things that happens is a P53 mutation happens. And that often causes the tumor to develop more aggressively. So we've seen the sequence of genetic changes. And as I said, P53 is generally thought to be knocking out the apoptotic or programmed cell death process. So that's the traditional view of lung cancer progression. And those mutations really do arise in the sequence. That's a fact. It's not uncommon in lung cancers for those mutations to arise in such a sequence. But with new cell state studies, what's happened is people look at the KRAS mutation happens first. And as I said, you get a little bit of a growth, but not an aggressive tumor. But when you can look at the cell states, what you see when the KRAS mutation arises is that the cells start transiting through a variety of cell states. Many of those cell states play an important role later in tumor progression. But at this stage, they're just there, and they're starting to appear as the precancerous cells are starting to transit through these different cell states. And then a P53 mutation arises, and the cells then begin to express a much wider set of cell states. Again, the sort of cell states that will eventually be important in tumor progression. Now, P53 is interesting because it plays an important role in cell state expression within the lung. In normal lung development, there's a precursor type of cell that is a developmentally flexible type of cell. And that precursor cell differentiates into the cells that line the surface of your lung, where gas exchange occurs. And the cells that line the epithelial surface of your lung are terminally differentiated and relatively stable, whereas this precursor cell is mobile and expresses many different cell states, transits between cell states, and can survive a variety of situations. And what we see when the tissue in the lung is damaged is that there's a de-differentiation back towards this precursor-like state. And we see that in the expression then of these more aggressive cellular state, which is important in cancer progression also. So wound healing and cancer progression both associate with de-differentiation, getting these earlier cell state forms. And what happens is that the knockout of p53 tends to cause de-differentiation of those terminally differentiated cells, because in normal development, p53 expression causes the differentiation of the epithelial surface, terminally differentiated surface. So you knock out p53 and you have a tendency to move back towards the more primitive de-differentiated form. So we see this de-differentiation once again. Now, a subsequent study on this same system in the lung looked at the different cell states that were involved. And I mentioned that when the KRAS mutation comes up, the cells start to transit between different cell states. And when the p53 mutation comes up, which is now we're moving on to cancer progression, we see that many different cell states are present and they're transiting between each other. And what the study did was measured the transition probability from one cell state to another. So you get sort of a transition or matrix or network describing how the cells are transiting between different cell states. And there were, I can't remember, 10 or 12 different cell states that were easily identifiable. And interestingly, there was a particular cell state that was kind of at the nexus of this network that could transit to many other different cell state forms. And that particular high plasticity cell state, which is what the authors of this paper called it, was interesting because it expressed trophoblast-like characteristics. Trophoblasts are cells that make maternal tissue during pregnancy. And this is important for cancer because maternal tissues bring in blood supply, grow very rapidly, and so many of the characteristics of a tumor use trophoblast-like characteristics. And so this particular cell was de-differentiating and including trophoblast-like characteristics in its gene expressions and proteins. It expressed chondroblast-like state, partially. Chondroblasts make cartilage and connective tissue in your body, so it's a totally different kind of tissue. And it also expressed some of the tubular epithelium that's important in your kidneys. And so the authors described in their own words that basically this cell is expressing parts of different developmental programs. Now remember at the beginning of the talk when we talked about the goat, I said how developmental recombination was Mary Jane West Eberhard's argument for how novel traits arise. You take bits of different developmental programs that are flexible and you recombine parts of them in a new way and you get a coordinated new characteristic that arises in one step through developmental recombination. And in this study of lung cancer, this is exactly what they're seeing and exactly how they described it themselves, that basically parts of different developmental programs were being recombined and re-expressed in the single high plasticity cell, which sits at the nexus of the cell types, which will eventually form the tumor. And so that's the key point. And now I want to summarize the whole talk in relation to that key point, which is that it's very rare to see two different fields which are going through intellectual changes in how they understand the world. In evolutionary biology, we have this phenotypes first idea, which is not the majority view presently. Genes first is still the dominant view, but this phenotypes first view is gaining interest and attention. And the phenotypes first view, as developed by West Eberhard, emphasized very strongly this notion of developmental recombination. And now in cancer research, a revolution driven by technology, measuring proteins in single cells, is showing us the same thing. Developmental recombination is the origin of novelty that's really driving tumor progression. And this is the way that people working in cancer are describing it. And the people working in cancer who wrote this article, they don't know anything about the evolutionary theory and evolutionary biology. They came to this idea completely independently. And when you see the convergence of similar perspectives from very different starting points in different fields, that's often a signal that something interesting is going on. And so we're very lucky intellectually and living through one of those rare times where we're getting this convergence of conceptual points of view. And this is just happening. These publications are quite recent. So this is really a prediction from me that in the years going forward, we're going to see a convergence of points of view between these two different revolutionary aspects of evolutionary biology and cancer research. And we have the opportunity to watch this develop in real time. So that's my prediction. Thank you.